Dr. Chris Wozniak will be next. Chris is going to talk about future products. Chris was an agricultural researcher with USDA years ago, working on insect biocontrol and plant engineering. He's an expert on biotechnology, human health, and ecological risk assessment. He's been with BPPD for 16 years. And Chris currently serves as a division special assistant for biotechnology. So to kind of help us peer into the future, here's Chris.
Thanks a lot, Mike. And you know they hired me for my clairvoyance. So as we peer into the future, I want to state the obvious that a lot of the products that I'm talking about are very speculative because they are in the future. Some of the ones we can anticipate because we go to meetings, we talk to people, people present information to us, et cetera, ask questions. But in some cases, we really have to put our best scientific hats on and think about what is possible given the techniques that are evolving. First, an outline. One of the things I'll mention, and I think Bob alluded to this, the National Academy of Sciences put together a panel to look at future products of biotechnology. Their report was out in 2016. It's freely available online for download as a PDF. This, by the way, was funded by the 3 regulatory agencies, and this was in conjunction with the Coordinated Framework Update. This is one of the 3 sort of parts of that effort, was to ensure that a panel of experts of both legal and science background and regulatory background looked at these future products or the potential for these future products. I'll go through that briefly. I want to switch gears then and talk a little bit about a blight-resistant American chestnut that's, even though it's somewhat traditionally manufactured, it raises interesting legal and regulatory questions for the agency. And then talk a little bit more speculatively about synthetic biology and the products that might arise from there, including also genome-edited pips. And one of the reasons I say that these are speculative is that we have not seen any submissions to the agency yet. So again, things that we know about by talking to people and attending meetings, et cetera, as well as coordinating with our other regulatory colleagues at FDA and USDA APHIS. Then we'll talk a little bit about RNA interference pesticide products that are not within a PIP. We've already seen a little bit of this, so I'll cover that just briefly. And then last, but certainly not least, gene drives, as they may be used in both conservation and in agriculture, particularly for mosquitoes and for rodents. So with the National Academy of Sciences, Engineering, and Medicine report, again, it's fairly thick and detailed. We'll just stick to the highlights. So this first couple of slides I actually took from the panel members, and this is basically an outline of their report. They looked with a 5 to 10 year window. What can we, anticipate will be coming down the pike, so to speak. They also evaluated the current status of the regulatory system in the U.S. and then also spoke to areas they thought may need some attention or enhancement in the regulation. And also asking the basic question, are there novel risks presented by these novel products, some of which we can anticipate, some of which we can't, but are there differences in the risk? And short answer is not necessarily differences in the risk, but some differences in how we approach the assessment of that risk. So as I mentioned, this is an approach to look 5 to 10 years out. There were many public speakers that came and talked to this panel about products that they're working on. Obviously, some of these are things that are getting closer towards realization. People don't often talk about proprietary mechanisms in public until they're fairly certain that they're moving down the path either with patents or with applications. It also asked about, our current risk analysis system and basically is it adequate? So they're looking for potential for different risks that might be presented by these. And I would say by and large, there are not different risks, but some of them may be more complex to measure. And I'll get into this as I talk about the individual technologies that may require a little different approach to understanding them and reducing uncertainty. And then, of course, what kind of capabilities does the regulatory system have? What kind of tools does it have? What does it need or what it may need in the future? And so that's all included. And I want to again reiterate that this is about conservation measures, and agriculture. This does not include human drugs or medical devices. So one of the challenges that the report put forth was seeking federal research funding increases. And I think we could all agree this would be nice, particularly those that are risk analysis related. There is a biotech risk assessment grants program through the National Institute of Food and Ag that is funded by USDA agencies as part of a checkoff program. And certainly we are involved with the development of the request for applications for that program, as well as serving as panelists to review the proposals and to meet with the principal investigators who are funded to help them better understand the regulatory requirements. Hiring federal scientists with the appropriate expertise, again, maybe perhaps obvious, but something that we strive to do. We already have 3 of the speakers you saw this morning, actually 4 of the speakers are fairly new to BPPD, and they certainly bring with them a wealth of experience and their relatively young lives. I'm feeling kind of old up here when I look over at the youngsters, but they're very impressive. And that's one of the things that we strive to do through horizon scanning and through bringing in speakers, whether they're from industry, academia, wherever, as well as hiring the correct people that have the experience. One of the real tricky parts, I think the panel correctly identified, is the fact that with synthetic biology in particular, you could potentially be looking at organisms that don't have good comparators. So in other words, I'm thinking particularly of microbial species. They talk about microbial communities. There was one example given where one of the speakers talked about using microbes to chelate heavy metals as part of mining operations, which might be something that our Toxic Substances Control Act might cover with the group downtown here at EPA. Now, some of those organisms may be named species, but they've been so radically changed, even large chunks of DNA removed and replaced, rearranged, that they may or may not serve, based on what you can find in the literature, as a good comparator. So that would definitely be a challenge. In addition, going along with, again, somewhat with the coordinated framework update, this idea of statutory flexibility and also identifying what products would fit under which agency's statutory authority. You might think, well, since the original coordinated framework was published in '86, this would all be a done deal, but you again have to realize that that original coordinated framework really had a focus on microbes, and GE plants were sort of brand new at that time. So as things evolved, we had quite a few meetings with multiple agencies, including particularly our regulatory partners at both EPA, OPPT, as well as the USDA, APHIS folks, and FDA, both veterinary medicine and Center for Food Safety and Applied Nutrition, to try and nail down who regulates what and why. In part, but I would say the impetus came from this is the fact that now we and Biopesticides are regulating the GE mosquitoes that are intended for population suppression as of roughly a year ago. And omics was, again, a highlighted area, and I think it in part reflects the expertise of some of the panel members on this esteemed panel. Omics covers a variety of things. There's transcriptomics, basically looking at all the transcripts that come off of gene expression. There's proteomics, looking at the global environment of proteins present in a cell or a tissue or an organ. And there's also metabolomics, looking at all of the metabolites, the non-proteins, the things that are produced by the proteins, you know, the terpenes and the vitamins, and could even be some toxins involved. Those are the kinds of things that we have not had to look at yet because we've been dealing with single traits with defined activities, defined modes of action, like cryoproteins, for example. As we move into things that potentially affect the basic growth and development and even the habit of the plant, whether we're talking about manipulating phytohormone levels or doing other sort of drastic measures to create novel pathways through synthetic biology, those would be instances I think the panel is referring to where some metabolomics might be in order for us to better understand what else is potentially being produced along with the transformation that we're reviewing. The data commons concept was another main part that the panel referred to several times within the report. The idea is here to create public databases that are available for use by everyone and anyone. And these could include things to do with allergens. I know one of my colleagues just mentioned Allergen Online as an example from University of Nebraska, toxin databases, other sort of general databases where the information may not be product specific, but would still be of use to a variety of folks. And certainly more involvement of social and natural scientists. And while we have a fair number of natural scientists here at EPA, with things like gene drives, as I'll mention, social science may play an increasingly important role. And again, aligning the complexity of our regulation, or really the complexity of our risk assessment, to be commensurate with the risk that we see in the products developed. So again, I hate to pick on gene drives, but there's an example where things would be a bit more complex than, say, a single trait into a plant for insect resistance or disease resistance. And with the outreach issue, that's always an ongoing issue. We certainly strive to do our best, whether it's at national meetings, local meetings, through webinars like this, or the one that Mike mentioned in December of 2016, which is archived online. We're constantly striving to work more with folks, even things like some of us are judges at high school science fairs. I've been working with colleagues at FDA to help develop high school curricula that focus on biotechnology and things of that nature to enhance the understanding and get people to hopefully understand the need for some regulation. So there are conclusions, some of which seem a little bit daunting to say. We may well be overwhelmed by the diversity and types of products and the sheer number that we see in the future. We certainly strive to maintain ourselves to handle basically whatever comes at us. And we do this in part by horizon scanning. And this includes the things that I've mentioned in the outline, things like gene-edited pips, synthetic biology, gene drives, keeping abreast of what's going on so that we're able to proactively respond. And I do agree with them that we will have some difficult challenges with some of these products, no doubt about it, and I believe we're up to the task. Now I want to shift gears and talk briefly about the American chestnut. Once was the predominant forest species in the eastern deciduous forest. Roughly 1 out of every 4 trees was an American chestnut. And they were huge trees, and they dominated in many ways. Also influenced the community structure by massive nut production. We used to refer to as mass production. People would carry these to local stations, and for some of them, it might be their only income for the year. They, of course, had quite a spectacular lumber industry. I just stayed in a cabin a couple of weeks ago made of chestnut from the 50s. As this tree died out, it changed the economics and the community structure, both biologically and economically, in many of these areas. So this group at the State University of New York wants to bring this tree back. It exists only as stumps with little sprouts, which die back every few years as this fungus maintains its presence. And we're sort of excited about the possibilities of bringing this back. Now, the mechanism for this is kind of interesting. So the fungus produces a compound called oxalate, chemically a rather simple compound, but it's toxic, kills the tissues. Ahead of the fungus can grow into these dead tissues as a necrotroph and proceed to basically cause a huge canker and kill the tree. Their way of dealing with this is to engineer an oxalate oxidase gene from wheat. This enzyme basically degrades the oxalate into hydrogen peroxide and CO2. And the fungus still persists, but it doesn't do great. A lot of people would see this as almost like an antitoxin, and I borrowed this slide, by the way, from the developer, so some of this is his language, but I tend to agree that what we're seeing here, and a lot of people, I think, confuse the idea of a pesticide has to kill something. Well, it absolutely doesn't. You know, it can be repelling, it can be mitigating to some degree without being lethal. And in this case, this is a good example where the fungus survives and even sporulates on the resistant trees, as you'll see in a second. So on the left is the transgenic tree, on the right is its genetic sort of sibling or control. And you can see that there's cankers on both, but obviously the one on the right is far worse, and that tree will eventually die. The Chinese chestnut, which some of you might be familiar with, On the left there, again, it's tolerant. It gets a small canker, but it does survive. The transgenic one on the right does as well, and you can re-isolate the fungus from both. So I guess the question for the agency right now is, as much as we'd like to see this reestablished in the environment as it was, and really is a native species, the question still remains, is it really qualify as a plant-incorporated protectant, or is it something different? So that's something that our policy folks are dealing with. I'd like to just briefly talk about synthetic biology. It's hard to find a good standard definition for synthetic biology because not everybody agrees on what is synthetic, but I'll just mention that some of the examples, certainly cases where whole chromosomes are exchanged out of an organism, whether it's a yeast cell with just a handful of chromosomes or a plant. Much of the work has been done so far in bacteria, and in some cases you can create little cells that are functioning on just a paucity of genes compared to what they originally had. And you can, of course, engineer some of these to do interesting things, create entire new pathways that don't exist in that species or related species. So they're truly synthetic. As I mentioned earlier, trying to find a comparator. And when we do microbial risk assessments, we always ask, well, what is the species? What do we know about that species? How does it act in the environment? Where does it survive? What temperatures does it grow at? Some of that goes out the window when you have a truly novel new organism. The good news is that in some cases, this can solve some of our issues. Somebody asked earlier about potential for gene movement and horizontal gene transfer between bacteria, we know goes on fairly often. So if you're moving a toxin-producing gene into a microbe to kill some pest in the soil, but you're concerned that that toxin can move to another bacterium because the gene could move, you can create this organism such that it uses an entirely different network to just even produce its own proteins. You change the structure of the ribosomes. You used atypical amino acids that don't exist in nature. And so you've created an organism that really can't communicate anymore with its brethren. So it's like, somebody speaking Latin to somebody who's speaking Spanish. You have the occasional common word or note, but there's really not a conversation there. That's sort of the goal. And there are some of these been well defined in the literature. There's a dead man type construct that's commonly worked on. It's like a toggle switch. Shut the organism off. There's passcode switches, a little more complicated, but the basic idea is they respond through a circuitry mechanism to cues from the environment, and they won't persist, or even if they do persist, they can't really talk to their neighbors, so to speak. So interesting stuff that we look forward to. Now, in terms of gene editing, you're probably aware of CRISPR-Cas9. That's been in the news a lot. There are other mechanisms as well. We, again, have not seen any of these come through the door. So as to exactly how we will handle these from a risk assessment standpoint, we'll look at it on a case-by-case basis and see what that particular product has and whether it's a PIP or not. But the great news here, again, is that we can do things so much more precisely. I always use the analogy that previously we were using a jackhammer, now we've got a scalpel. So we can go in and change just a few nucleotides on one gene, whether that's to shut that gene off or to turn it back on. We could create a disease-resistant crop with that small of a change and save years of complex plant breeding, not to mention the money. And this is true of plants as well as there's an upcoming FDA seminar on using these techniques in animals. And this is really, I would say, truly revolutionary. Now, it'll reduce the uncertainty of a lot of unintended effects because, again, when you do crosses and things, whether it's after mutagenesis or transgenesis, there are a lot more changes to deal with. This is so far more precise that I think it'll allow us to focus the risk assessment even tighter. We do know that people have already created disease-resistant rice, for example, and wheat. We have not seen them come through the door yet, although our colleagues at USDA-APHIS have seen some of these. I use this just to point out that this meteoric rise in the CRISPR-Cas9 reflects a couple things. One is the system comparatively is easy compared to, say, for example, creating a zinc finger or a talin molecule, which can accomplish more or less the same thing. And there are people still using these. Just recently I saw a soybean produced with zinc fingers that is a great product. But this CRISPR is cheaper, faster, and easier, and that's going to bring a wider diversity of products because you'll have more people working with it. Academics that may not have the wherewithal to have a regulatory staff and have a large group of folks focusing on production can create some really phenomenal products using this mechanism. I won't say too much about RNAi here since my colleagues have covered it so well, but let me just point out here that the idea here is we block the conversion of a messenger RNA template being used to create a protein. So the fact that we have no protein, we have no allergenicity concerns. That's the first big plus. But in addition, these double-stranded RNA molecules can be sprayed through a nozzle or put into a bait, for example, for fire ant control. And they're far more stable. Those of you who've ever worked with RNA in the lab and you used to have to bake all your glassware and just pray that nothing happened that day to keep the molecule stable, these double-stranded molecules are far more stable than that. So another thing that we're looking at is the environmental fate. How long does it last in an aquatic environment, particular soil type environments, etc. But you can spray these, and we know from talking to folks, even though, again, we haven't seen a product, that many of them are absorbed into the plants. They can be used to take a otherwise herbicide-resistant weed and turn it back into a susceptible weed by blocking the enzyme that's providing the resistance. Safe, they are broken down, as you probably all know. Nucleic acids are depurinated in the stomach, and so hydrochloric acid is not a friend. Not an issue there with uptake, and you can make these very specific because you can theoretically target any gene in the plant. You think about that in terms of insect resistance. We already have one product that's coming online here commercially, hopefully in another year or so. We've got the potential for adding all kinds of RNAi-based genes where we target one or more genes within a target pest and inhibit that pest's ability to either produce eggs or to feed or digest or whatever it happens to be. It's far more fruitful, and then this is what I used to do. You go out and dig in the soil for the next BT and trying to find out what that BT has in it. It's a bit of a numbers game. This is far more precise and directed, and I'm excited about it. Last but not least, I'll talk about gene drives. Again, you've probably heard about this in the news. I think it's even on the cover of Time magazine. People talking about this ability for skewed inheritance. So typically, a gene, as we all know, is roughly a 50% chance of getting into a particular egg cell or a sperm cell. With this type of gene drive system, and I should mention there are some of these that are naturally occurring. This is not totally man-made. If you look on the upper left here, the diagram A, the blue represents our modified gene, the one we want to drive into the population. And again, 50%, one half of the progeny get it, one half don't. With the gene drive, basically this element recognizes that its sister chromatid here does not have the gene and automatically copies itself. So now in the bottom C, you can see when in this pedigree chart that we ultimately end up with all the progeny inheriting the gene. So depending on what that gene is, it could either be a lethal gene, which will eventually drive itself to the end because there'll be nothing left to reproduce, but you could drive a population down rather quickly. You could use genes that are going to stop, say, malaria transmission in a mosquito. Mosquitoes survive, but they can't move the malaria parasite. Those are the kinds of things that people are looking at seriously. I should mention, too, that these are currently under a moratorium. It's a voluntary moratorium by the scientists who were researching these in countries all over the world, such that at some point, it's hard to say when, through public debate, regulation, discussion, etc., these could be applied for conservation measures as well as agricultural measures, but it's certainly going to require a significant outreach and public debate, etc. And this is particularly, I think, where the Academy is mentioning about social scientists being involved in these kinds of decisions, because in theory, you could design a gene drive to, say, wipe out the yellow fever mosquito permanently off the planet. And so that's a major shift from depressing a population with a spray or a BT Israeliensis in a pond or whatever. So this is sort of new ground, so to speak. As a matter of fact, tonight I'll be on listening in on a call or a meeting actually in Perth, Australia, where they're engineering rodents to basically wipe out rodent populations on Pacific islands that have wiped out a lot of birds. And this group has got some people here from the States, North Carolina State University, Texas A&M, New Zealand, Australia, Britain, you name it. And they're moving forward with this. So this is not something that's going to happen 10 years from now. There are people almost ready to go, not quite there. And so this is something that the agency, I have a feeling, will be facing. We still need to figure out exactly who will regulate what in this realm, but certainly something that has been the topic of discussion. In summary then, just mentioning the obvious challenges to the regulatory system by all these types of products. We may need to adjust our risk assessment process in some instances. Certainly for the gene drives and some of these things, we may require more mathematical modeling and probabilistic risk assessment to run things on the computer prior to any field release. And certainly, looking forward to things like more of these double-stranded RNA-based products where the safety profile looks excellent and they seem to have, at least what I'm hearing from folks, good efficacy and good specificity. And again, these gene drives, that's going to be a topic of debate. Don't know how long it's going to be, but certainly sort of the wolf's knocking at the door, as they say. So we'll be discussing these. I think the rodent example is probably one of the first. There are plenty of others out there. Mosquitoes, for example, where folks are talking about the Gates Foundation, for example, is invested in malaria reduction in Africa. So these are the kinds of things I think that really would be game changers. And with that, I will close.